Hello everyone, I'm Mohammad Ahmed Khan. Today we will discuss about protein. So the first point is the protein is basically made up of different type of amino acid. That's why it is known as heteropolymer. So the first point about the protein is it is heteropolymer of amino acid. And why it is heteropolymer? Heteropolymer is basically because it is made up of different amino acid, different combination of different amino acid. That's why it is known as heteropolymer of amino acid. The second point about the protein is it is involved, the protein synthesis is takes place basically on the ribosome. The next point about the protein is its function. Like its structure, it perform diverse role. So its function is also diverse. The basic function is basically the very first function is almost all the enzymes are made up of protein. You may heard about the pepsin, which is involved in the digestion of protein. Is enzyme is a protein. Trypsin is also a protein digestive enzyme. Amylase is a carbohydrate digestive enzyme. All enzymes, almost all the enzymes, are made up of protein. The second basic function of protein is hormone. Many hormones are made up of protein. The next function of protein is antibodies. The antibodies which provide protection against any antigen or any infection are also made up of protein. There are five different type of antibodies like IgG, IgE, IgM, IgA and IgD. All these antibodies are made up of protein. The next basic function of protein is transportation of substances. Transportation of substances like, for example, GLUT4 protein. GLUT4 protein, which is involved in the glucose transportation, is also a protein. The next function of protein is structural role. Its structural role. You may see in the plasma membrane. The plasma membrane has two different type of proteins like integral and the peripheral. And in the plasma membrane, it perform as structural role. The another structural role, maybe you may heard about the collagen protein, which make intercellular ground substances, is also a structural protein. So these are the basic function of protein. Now we move to the structure of protein. Structure. So the protein has four structural level named as primary structure level, secondary structure level, tertiary structure level, and the quaternary structural level. So we will see these four level of structure one by one. So the very first level of structure is primary structure. Primary structure. So the primary structure level is nothing but it is the linear arrangement of amino acid. In the primary structure level, The neighboring amino acids are linked through the peptide bond. So I will try to explain it with a very simple reaction. So this is the first amino acid, and this is the second amino acid. So the hydroxyl group of this carboxylic group and the hydrogen of this amino group react with each other, and there is dehydration, there is release of water. So in this way, the neighboring amino acid are linked through this bond, which is known as peptide bond. So in this way, there is linear arrangement of amino acid, and this is known as the primary structure level. The left hand is known as amino group, and the right hand is known as carboxylic end. And it is the first amino acid, and this is the last amino acid. So in this way, the different amino acids are arranged and linked with the help of peptide bond. The primary structure is very important from the point of view of the further structure level of protein. If there is disturbance in the primary structure, then the resultant protein is also distorted. So the point of view of structure of protein, the primary structure is very important. Now, the secondary structure. So, in case of secondary structure, the primary linear chain which we get in the primary structure level undergo further processing, and it produce alpha helix or beta plated sheet. Alpha helix or beta plated sheet. So there is alpha helix, this is alpha helix, and in case of alpha helix, the there is intramolecular intramolecular hydrogen bonding is present between first and fourth amino acid. And the example of alpha keratin protein is keratin protein. The alpha helix is found in keratin protein, which is present in our hair and nails. The another one secondary structure is beta plated sheet. In case of beta plated sheet, two polypeptide chains are arranged in parallel manner, and here we get intermolecular hydrogen bond. The beta plated sheet may be parallel or anti-parallel. If the amino group of two chains and the carboxylic group of two chains are on the same side, then it is known as parallel beta plated sheet. And if it is on different side, then it is known as anti-parallel beta plated sheet. The beta plated sheet is basically found in fibroin protein which is present in cell. So this is secondary structure. Now we will move to tertiary structure,